Good morning. Thanks to Francisca and Lara for this invitation, and thanks to Johannes for his very instructive presentations. I hope that my presentation will support with more practical elements the session. Reporting in medical genetics is an activity that involves the combination of genotyping, so laboratory findings, and clinical data. Reporting in medical genetics is a clinical act, is a medical act aimed to support, to demonstrate, or to exclude a causation between the variants, the identified variants, the genotype, and what the clinician sees in the clinics. In the context of current knowledge that is evolving, that is changing, and that could be summarized in a term that is human variome. Working in labs means that for every medical genetics report, we need to respond, to face at least three main questions. The question one is that the gene in which the laboratory found one or more variants of interest is really associated with the phenotype observed by the clinician according to the current knowledge. Question two is the supposed molecular pathogenesis, and on this argument, the presentation by Johannes is absolutely illuminating, is the supposed molecular pathogenesis that we hypothesize for the variant of interest is in line with the genotype-phenotype correlations that currently we recognize according to the literature. And the question three is the causation that we suppose linking the genotype to the phenotype supported by enough evidence? If for a positive report, we need an assertive positive response to all of these three questions. To address the third question, the American College of Medical Genetics in 2015 published their criteria. They are 28 criteria organized in 16 criteria of pathogenicity and 12 criteria of benignity. Their combinations identify the variant class that can be pathogenic, likely pathogenic, of uncertain significance, likely benign and benign. In the original paper in 2015, the approach was heuristic. So the authors proposed the various, more typical combination of criteria that could be assigned to every clinical class of variants. 3 years afterwards, a working group within the ClinGen initiative proposed a Bayesian approach that Johannes showed in a previous slide. They proposed that the attribution of every single class can be defined by a posterior probability cutoff. They assigned a specific weight, so a specific posterior probability to each level of strength. So the combination of these posterior probabilities define the variant class. Likely pathogenic and pathogenic variants that might justify the phenotype are true positive results because they demonstrate a causation, support the causation, a causation between the genotype and the phenotype. But pathogenic and likely pathogenic variants could be still some kind of positive results if they fall in actionable genes, also in case of genes that are not related to the phenotype. If the patients gave their her or his patient consent to secondary findings. Sometimes pathogenic, likely pathogenic variants in their current form do not completely support the phenotype, so they might be intended as incomplete results that need, for example, the identification of a second mutation in a recessive gene or the demonstration of the phase in presence of two heterozygous variants, again in a recessive gene in the absence of the parental DNA. Variants of uncertain significance are inconsistent results that might switch to positive results if we can generate additional data supporting their pathogenicity. But on the other side, there are many variants of uncertain significance that are don't have really probably real clinical role. So reporting variants of uncertain significance is in the middle between the need to stimulate clinicians to give more additional information of additional biological samples for further studies in order to demonstrate the hypothesis that the variant of uncertain significance is the cause. But on the other side, they might increase anxiety and anxiety for the families, but also a confusion to the specialist, especially if he is not so familiar with medical genetics. So managing variants of uncertain significance force the laboratory staff, but also the clinicians, in recognizing the need of close interactions between the lab and the clinicians. Because as you can see, the process of reporting is a deductive approach that need some activities that can remain within the laboratory, but others, such as the weight that we can give to the phenotype, collection of additional samples from the family, or for the proband for functional investigations, and segregation data, need absolutely that the clinicians recognize the complexities of the laboratory activities. Criteria associated with allele frequency are usually assessed first because the Mendelian rules applied to rare phenotypes easily make easy the definition of specific cutoff, especially for conditions with specific Mendelian inheritance patterns and food penetrance at an early age. Factors influencing the cutoff are allele frequency, the disease frequency, genetic heterogeneity, and penetrance. Usually, there is a direct relationship between allele frequency and disease frequency, and an inverse relationship between allele frequency, genetic heterogeneity, and penetrance. The higher is the allele frequency, the disease frequency, the higher the allele frequency is. The higher the genetic heterogeneity and penetrance are, the lower the frequency cutoff for pathogenicity is. PVOS1 criterion is the criterion that is associated with variant predicted to cause null alleles. But in the absence of transcriptional and post-transcriptional data, there are many factors that might influence the real level of strength that you can attribute to this criterion. Currently, you can use the AutoPVS1 website in order to predict the most likely level of strength to be associated with the specific PVS1 criterion. There are additional criteria, especially for missense variants. There are at least 5 criteria that can be assigned to these variants according to their relevance in public repositories and PubMed. In order to assign these criteria, the American College of Medical Genetics does not establish rigid rules, so every laboratory needs to develop a specific internal pipeline. In the development of these pipelines, I suggest to consider some red flags. For example, the way to give to the number of annotations in ClinVar, how to manage conflicting data, the possibility to use the star system, and sometimes the need of manual review of the PubMed citations that you might retrieve from the ClinVar API file. Again, for missense and for a restricted number of variants of variant type, the American College of American Genetics proposed a set of criteria to assign to in silico predictions. In silico predictions are not strong factors influencing the final interpretation of the variants, but they might support the interpretation, especially in presence of private variants and in the absence of the possibility to carry out functional investigations. But also in this case, we have many caveats to consider. Currently, available tools are applicable especially for missense and for missense variants, structural variants, and variants predicted to alter the splicing. We should to avoid to refer to a single predictor. You need to consider the right predictor for the right predictions. And again, I suggest to consider a training period, especially in order to select the right combination of tools for the application of these criteria. For missense and in-framing DELS variants, again, especially in case of private variants, there are 5 criteria that can be assigned according to specific molecular features. The PM1 is related to variants falling in hotspot domain or critical protein domains. The PM4 and BP3 criteria is associated with in-framing DELs if they fall outside or within repetitive regions. And PP2 and BP1 criteria are associated to missense variants falling in genes with a high rate and a very low rate of deleterious missense variants according to public repositories. Segregation data are determinant, especially for Mendelian conditions with full penetrance. The PP1 criterion that can be seen with different level of strengths stands for the co-segregation of the phenotype with the genotype within the family. The number of meioses demonstrated co-segregation influence the strength of the criterion, according to specific proposal that you can find in the ClinGen website. On the other side, there are criteria strong benignity criteria that can be used in specific circumstances in order to downgrade the relevance of the variant in case of non-segregation of the variant in at least one clinically affected family member, and segregation from an unaffected parent or the presence in another relative in case of full penetrance at an early age. Here a summary of the impact of the various combination of these criteria to the final posterior probability of pathogenicity. Segregation is also useful in presence of multiple variants at the same gene, for recessive genes and also for autosomal dominant genes. Usually, NGS molecular testing generate multiple candidate variants in different genes. In this case, close collaboration with the clinicians and the integration in the family could help also in discriminating variants among falling in different genes, and therefore the attribution of the BP5 criterion to one or more variants. Since 2015, there are at least 3 criteria that are strongly influenced by the specificity of the phenotype. The PP4 criterion is attributed to the phenotype in terms of close relationship of key clinical features and the literature. The American College of Medical Genetics criteria do not offer rigid rules for the application of the PP4, so every laboratory should develop internal rules. For example, this is our approach to the use of PP4 in Marfan syndrome. Phenotype specificity can influence also the de novo criterion. This is the different impact of the combination of these criteria to the posterior probability of pathogenicity. Finally, there are 2 specific criteria associated with the functional data supporting pathogenicity. In routine diagnostics, we can distinguish 2 broad groups of variants: variants altering the splicing and structural variants altering specific cellular functions, specific pathways. Splicing variants are easier to study because real-time PCR and whole transcriptome sequencing could support the laboratory in this issue. Structural variants could be investigated only by specific cellular tests. This is an example of real-time PCR in testing a candidate splicing variant in vascular ADS. This is an ex vivo cellular study in a case of spona-string dysplasia and double missense variants on the tonsil gene. And this is the luciferase assay used in supporting pathogenicity of a missense variant in the TAB2 gene that fall in the same protein domain in which we found many nonsense variants. This is the last slide and a summary of the workflow that one could follow in the attribution of the various ACMG criteria from the VCF5 to the generation of the report. This work could be carried out properly only if the clinician recognizes the relevance and the complexity of the laboratory activities, because in the absence of close collaboration, the presence of one or more variants of uncertain significance is the most probable content of the clinical report. Thank you very much.